So earlier on, we talked about metabolism, the anabolic and catabolic reactions in the cells. When we talk about that, it's not as simple as metabolite A turn into metabolite B, or lipids turns into fatty acids. So there are actually a big complexity in it. So there are a lot more reactions that involve in between in order to produce those final products. So these are just the fractions of what are the metabolic pathway or metabolic map. So for example, this one is in mammalian liver metabolism. So just imagine this is, in a cells. So what we have here, for example, the pentose phosphate pathway. This is the TCA cycle. All right. So in E. coli, for example, it has about maybe 5 to 6 million nucleotides, and then those nucleotides encode for maybe 4 to 5,000 genes that encode for proteins, and those proteins are actually the one that involved in catalyzing the reactions that convert one molecule to another molecule. So now you can predict that what we're going to talk about in the next topic, that is the enzyme that catalyzing all the chemical reactions in life. So in metabolism, not just energy that is important, enzyme is also important. So before we discuss further on how cells produce energy, so let us consider about the principal properties of a group of proteins called enzyme. So these enzyme or these proteins are involved in all of biologically important chemical reactions. All right, so it is important to understand that a cell's metabolic pathway, which is the sequential chemical reactions in the cells, are determined by its enzyme. All right, of course, an enzyme is determined by the genetic makeup of the cells, the DNA. And enzyme catalyze the reaction for specific molecules, which is called substrates, and the substrate is transformed into a new substance called product. For example, here we have sucrose is a substrate, and then we have sucrase, the enzyme that degrades sucrose into glucose and fructose. So sucrose is a disaccharide. So sucrase will cleave the disaccharide into glucose and fructose. And this enzyme sucrase is specifically identify the substrate sucrose. So it's like a lock and key phenomena or concept. So only sucrose can open up or react with this sucrase. Then that sucrose is then turned into a product depending on the type of the reactions that the enzyme will do, right? So before we go into detail about enzymes, so let us know about what are the characteristics or features of enzyme. Okay, number one, enzyme are mostly composed of protein. All right, so most enzymes are protein, but not all protein are enzyme. Okay, and an enzyme may require cofactors that aid in the function of the to add in the function of the enzyme. Number two, enzyme act as organic catalysts to speed up the rate of cellular reactions. So we have only sometimes when we want to dissolve sugar, for example, in water, we need heat. If we put heat, then the sugar will dissolve faster, right? But in this case, we don't use those physical treatment or physical factors or chemical factors, but instead we use enzyme as organic catalyst to speed up the reaction. Number two, it lowered down the activation of energy required for chemical reaction to proceed. We're going to look into that in detail later. And then enable metabolic reactions to proceed at a speed compatible with life. So everything happen in cells are very fast. For example, if we have E. coli that divide or double every 20 minutes, just imagine in that 20 minutes, it has to do all the reactions in the cell to break down the glucose, to produce nucleic acid, to produce proteins, to produce enzyme, to produce lipid, to produce its structure, and so on within 20 minutes. Next, so enzymes have unique characteristics such as shape, specificity, and function. So it all depends on the structure or the build up of the enzyme. The amino acid to be specific, and then that amino acid will turn into primary structure, secondary structure, how it actually folded into 3D structure. So all are unique to that enzyme. Then it provides an active site to target molecules called substrate. It has an active site where the substrate will bind to it, then the reaction will start. Next, enzymes are much larger in size than the substrate. Okay, enzymes associate closely with substrate but do not become integrated into the reaction product. So the enzyme will not be changed. All right. They are not used up or permanently changed by the reaction. Enzyme can be recycled, can be used over and over again. So thus function in extremely low concentration. So although the cells only have very low concentration, the reaction still can occur. And enzymes are greatly affected by temperature and pH. They are very sensitive. How enzymes accelerate reactions, so how enzymes speed up the reactions. So before we go into that, so let's first talk about the collision theory. Chemical reactions occur only when chemical bonds are either firm or broken, right? Either the big biomolecules is broken down or two molecules become one. For reactions to take place, either atoms, ions, or molecules must collide. So the collision theory explains about how chemical reactions occur and how certain factors affect the rate of those reactions. We say that enzymes will lower down the activation energy. So what does activation energy? So what is activation energy? So activation energy is the collision energy that is required for a chemical reaction to occur, which is the amount of energy needed to disrupt the stable electronic configuration of any specific molecule so that the electrons can be rearranged. So one way to increase the reaction rate, how fast is the reaction, so we can actually do that by increasing the temperature. So when at high temperature, molecules will move faster. So heat will increase both the frequency of collision and the number of molecules that attain the activation energy. Also, the number of collision also can be increased by pressure, by putting pressure. So when pressure is increased, or also when reactants are more concentrated. If you have more reactant substrate, then you get faster rate of reactions. So in living systems, so enzyme will increase reaction rate without the need or without the increase of temperature, because enzyme is heat sensitive. So a significant temperature increase will destroy the cellular properties of the proteins. so that it won't be functional. So enzyme will function. Enzyme function to speed up the biochemical reactions at a temperature that is compatible with the normal functioning of the cell. Okay, so let's take a look at this graph. Okay, so we are talking about the reactants here. AB is one molecule, all right, and then so we're looking at the reaction of AB molecule converted into product A plus B. Okay, two products. Okay, so this graph shows the progress of the reactions both without the enzymes here, reaction without the enzyme, and with the enzyme. Now we can see that the reaction activation energy needed is much higher when there is no enzyme. All right. So again, you have to refer to this definition, what is activation energy. And when the enzyme is there, then the activation energy is much lower. So this just means when in the presence of enzyme, more molecules of reactant AB will reach to the activation energy, or will possess activation energy needed for the reactions to be proceeded. Okay, therefore AB will be converted to the product. Okay, now let's take a look at the enzyme reaction. So what is the mechanism of enzymatic action? So let's take a look at here. We have enzyme, we have substrate here, enzyme, and then we have substrate, we have enzyme substrate complex, and then we have product formation, and then finally back to the original form of the enzyme. Okay. So the mechanism. First, you have substrate here. The surface of the substrate must first contact a specific region here. So you can see the substrate form. The form of the substrate is similar to the form of the enzyme, just like the lock and key. If you don't have the correct key, you will not open up the lock. So this region or this site is called as active site. Okay, once substrate is now in the enzyme active site, so there is this complex called temporary enzyme substrate complex. Okay, is formed. So in this formation, the enzyme will orient the substrate into a position that increases the probability of reaction, which enable the collision to be more effective. Okay, then the substrate molecule is transformed by the rearrangement of existing atoms, the breakdown of the substrate molecules, or a combination with other molecule substrate, and then the substrate now become product of the reactions. So the product of the reactions are now released from the enzyme molecule because they are now no longer fit to the active site of the enzyme. Then after release of the product, then you have the enzyme enzyme here that is unchanged. You can see it is still similar form. Now this enzyme is free to react with other molecule substrate, other substrate molecule. Enzymes have specificity of a particular substrates because of the three-dimensional shape of the specific amino acids of the active sites. Also for the overall structure of the enzyme, and the active site fits with the substrate as somewhat like the lock fits with the key. All right, as I explained earlier. The unique configuration of each enzyme enables it to find the correct substrate from among the diverse molecules in a cell. So just imagine in a cell we have thousands of substrate or metabolites. So the enzyme will recognize specific or will act on specific substrate only. However, the active site and the substrate are quite flexible. It can change shape somewhat as they meet together more so that they can fit together more tightly. Okay. Enzymes are efficient. See the optimum conditions that can catalyze reactions at the rate of 10 to the power 8 to 10 to the power of 10 times higher than the reactions without enzyme. So this is like billions or trillions of times much higher. And the turnover number, the maximum number of substrate molecules that an enzyme can convert to product each second, in 1 second, how much? So between 1 to 10,000 product formation, or can be as high as 500,000. So enzyme is super efficient. So otherwise you don't get turnover time or doubling time of E. coli for 20 minutes only. So how do you recognize enzyme? Because of the name. Okay, so normally enzyme names end with A's. Okay, at the back you have A's. And they are enzyme. There are 6 groups of or 6 classes of enzymes, and this is based on the type of chemical reactions that they catalyze. Enzymes. So what are the 6? They are oxidoreductase, which the reaction for catalyze the reaction for oxidation reduction, transferase that transfer of functional groups, for example amino group, ester group, or phosphate group. So they are transferase, hydrolase that involve in hydrolysis or addition of water. For example, lipase, sucrase, transferase. Just now we have kinase, transfer of phosphate, oxidoreductase, cytochrome oxidase. Okay, and then we have lyase that removal of group of atom without hydrolysis. For example, oxalate dehydrocarboxylase. We have isomerase that rearrange atoms within a molecule. You know, you have similar formula but different in structure. And then lastly, ligase, the one that join 2 molecules together. For example, acetyl-CoA and acetyl-CoA synthetase. So the enzyme name the name within each of the major classes are named according to the most specific type of reactions that they assist. So if you can see that lipase work for lipid, whereas sucrase work on sucrose, and so on. Enzyme component. So what is the enzyme component that make it functional? All right. So although some enzymes consist entirely of proteins, so most of the enzyme consists both a protein portion, and plus with the non-protein component. So most enzymes are actually apoenzyme that contain only protein, and plus cofactor, which is the non-protein. So if the cofactor comprise of organic molecules, it is called as coenzyme. And if it's the cofactor, and it is termed as cofactor if it is inorganic compound, such as iron, zinc, magnesium, calcium, and so on. So if the cofactor, the cofactor is organic, you call it as coenzyme, whereas if it's inorganic, you call it as cofactor. So many enzymes require apoenzyme plus cofactor to be active. All right. So apoenzyme alone, which is the protein portion alone, it is inactive. But when coenzyme is there or cofactor is there, then only it can be active towards the substrate. Okay. And this complex of apoenzyme and coenzyme is called holoenzyme, whole enzyme. And this holoenzyme is the active form of enzyme. So we're going to look into what is the function, or what are the functions of the cofactor or the coenzyme. Okay, so we know that for bacterial or microbes to grow, if you in the media of the growth media, you need to add in some of the trace metals, trace elements. So what are trace elements? So the trace elements include the the iron, the ferric, the magnesium, and so on. So those are important for the as the cofactor for enzyme function. All right. So one of the function is of cofactors is to help catalyze the reaction by forming a bridge between the enzyme and the substrate. Okay. For example, magnesium will form a link between the enzyme and the ATP molecule. Also, cofactor assists enzyme by accepting atom removed from the substrate or donate atoms required by the substrate. Also acts as electron carriers in enzymatic reactions, either to remove or donate electrons. Okay, so those are the function. And let's see the where does this cofactors derive from. So coenzyme, for coenzyme derive from vitamins. You can all these are the coenzyme NAD plus, NADP plus. So those are from the B vitamin niacin, nicotinic acid, and these NAD plus, function as electron carriers. Okay, in the cell, so carry a lot of electrons, and in the end, you will form energy from the oxidative phosphorylation. FMN, flavin mononucleotide, flavin adenine dinucleotide, FAD, derived from B vitamin riboflavin, function as electron carriers. Coenzyme A, derived from pantothenic acid, vitamin B. Okay, so that's all about the enzyme and the coenzymes.